This method can answer key questions in the field of mechanobiology, such as how forces are transmitted and sensed within cells as well as between cells and their environment. The main advantage of this technique is it allows access to the molecular scale information regarding how proteins respond to mechanical forces inside the native context of a living cell.
Though this method has been applied specifically to studying the focal adhesion protein vinculin, it can also be applied to other mechanically sensitive proteins, such as talin, cadherins, or nesprin.
Visual demonstration of this method is critical, as proper imaging setup is difficult but necessary for the success of the analysis.
Begin this procedure with stable expression of the tension sensor construct in the desired cell type as detailed in the text protocol. To prepare substrates for cell seeding, acquire 4 35mm glass-bottomed dishes. Working in a cell culture hood, in a 15 ml conical tube, make 4 ml of 10 micrograms per milliliter fibronectin in sterile phosphate buffered saline solution, or PBS. Gently invert the tube once to mix, and let the solution sit for 5 minutes in the cell culture hood. Then pipette 1 milliliter of fibronectin solution onto each glass-bottomed dish. Leave the fibronectin solution on the dishes for 1 hour at room temperature or overnight at 4 degrees Celsius. Then aspirate the fibronectin solution and rinse once with PBS. Leave 1 milliliter of PBS on the dishes until the cells are ready for seeding. After counting the cells, seed 30,000 cells onto each fibronectin-coated glass dish with the appropriate complete media for a final volume of 1.5 milliliters. Allow the cells to spread for 4